To reverse the biological age of a living cell, researchers have to activate genetic pathways that are inherently oncogenic. In 2006, Dr. Shinya Yamanaka identified 4 specific transcription factors, OCT4, SOX2, KLF4, and CYMY, that could revert mature cells to an embryonic state. Transcription factors are proteins that control which genes are turned on or off by binding to specific DNA sequences. While powerful, continuous expression of these factors inside a living body is a biological dead end. Cells eventually lose their mature identity, which leads to dysplasia, the formation of chaotic tumors called teratomas, or total organ failure. This failure occurs when a cell crosses a specific molecular threshold, the expression of the genes NANOG and LIN28. These markers signal that a cell has transitioned irreversibly into full pluripotency. Because full pluripotency destroys the structural identity of the tissue, it is incompatible with in vivo healing. Regenerating damaged organs requires an approach that stops well before this threshold. The strategy to bypass this trap is known as partial reprogramming. By turning these factors on for a precisely controlled, transient duration, researchers can reset age-associated epigenetic drift. The process is halted before the cell erases its underlying somatic identity. This graph illustrates decoupling. Epigenetic age drops, returning to a younger profile, while somatic identity remains fixed. The cell rejuvenates without becoming a stem cell, entering a plastic state primed for repair. This also downregulates SASP, pro-inflammatory cytokines from aging cells that damage tissue. By decoupling age from identity, partial reprogramming changes the strategy for regenerative medicine. Instead of trying to replace cells, the focus is on enhancing the endogenous repair capacity already present in the tissue. Eukaryotic aging is defined by a progressive loss of epigenetic fidelity. As cells age, they lose these regulatory markers, leading to the erosion of the information required to maintain tissue function. This timeline represents the inducible changes to the epigenome, or ICE, model. By inducing non-mutagenic DNA breaks, researchers can accelerate aging purely through epigenetic degradation, such as the loss of H3K27AC. H3K27AC is a histone modification that marks active enhancer regions in the DNA. To reverse this decline, researchers use a cyclic induction protocol, turning the factors on for 2 days and off for 5, repeated weekly. During the on phase, the cell restores youthful DNA methylation patterns and critical heterochromatin marks like H3K9ME3. H3K9ME3 is an epigenetic tag that keeps specific sections of the genome tightly packed and silent. The off phase stops the process just before the cell activates NANOG. This effectively applies the brakes before the cell reaches the point of pluripotency. This precise dosing protocol chemically neutralizes the pluripotency trap, allowing for a safe, long-term reversal of age-associated decline. Certain mammalian tissues, such as the adult heart and the central nervous system, have virtually no intrinsic ability to heal themselves. These organs lack resident stem cell pools and have extremely low reprogramming competence, meaning their mature cells are naturally resistant to epigenetic remodeling. In the visual system, repair requires highly localized delivery. Researchers use an AAV factor to deliver 3 of the factors strictly to the retinal ganglion cells, while specifically omitting the dangerous oncogene known as c-MYC. This schematic shows the biochemical requirements for repair. Axonal regrowth in the retina is dependent on TET2 enzymes, which must physically cleave off methyl groups to demethylate the DNA. If these TET2 enzymes are blocked, the regenerative effects are lost. Without active demethylation, the cell cannot overcome the intrinsic barriers to axon regrowth. In the adult heart, the barrier is even higher. Because cardiomyocytes are so resistant to change, initiating regeneration requires vastly intensified and prolonged expression of the Yamanaka factors. This intense induction forces mature cardiomyocytes to disassemble their sarcomeres, their internal contraction machinery, and re-enter the cell cycle in a fetal-like state. For these restricted organs, partial reprogramming acts as an exogenous driver, forcing open a window of plasticity that the tissue cannot generate on its own. This is a sharp contrast to highly competent organs like the liver and intestine, which are naturally regenerative. In these tissues, severe injury naturally forces mature cells to de-differentiate into transient regenerative progenitors, or revival stem cells, to rebuild the organ. Under normal conditions, shown on the left, immune cells like macrophages trigger this de-differentiation by secreting inflammatory signals such as interleukin-6. However, partial reprogramming follows the bypass pathway on the right. It reaches the same regenerative state without requiring any external inflammatory trigger. The factors directly upregulate PTGS1, the gene for the COX-1 enzyme. This allows the cell to autonomously produce its own prostaglandin E2, or PGE2. This cascade activates the EP4-YAP axis. This results in the same regenerative progenitor state, but it is achieved entirely within the cell, bypassing the need for systemic inflammation. This state requires a metabolic trade-off. The liver temporarily represses high-energy functions, like bile acid synthesis, to prioritize the metabolic demands of rapid proliferation. In competent tissues, partial reprogramming acts as an autonomous amplifier. It triggers the body's own repair programs while avoiding the damaging side effects of inflammation. The ultimate success and clinical safety of in vitro reprogramming is dictated entirely by a tissue's intrinsic reprogramming competence. This matrix shows the two distinct delivery architectures. Restricted tissues, like the heart and retina, require localized AAV vectors, while competent organs, like the liver, can be managed through systemic cyclic induction. Mapping these divergent architectures is essential to preventing organ failure while maximizing the regenerative response. Rejuvenation does not require a total reversion to embryonic states. It only requires the targeted erasure of accumulated epigenetic memory. By mastering spatio-temporal control and metabolic decoupling, medicine moves past the blunt risks of pluripotency toward the precise engineering of mammalian organ regeneration.